Well, thank you very much for those kind words, and also thank you very much for the introduction to come, for the invitation to come and speak to you here today. It's a great privilege to address you. Now, what I'm going to say overlaps with what you've just heard from Professor Yoshida. I'll give a general introduction to this topic, and then I'll go on to talk about how we can use the basic knowledge of how the ATP synthase works to medical benefit for the benefit of mankind. Now, let me start at the very beginning and repeat some of the things you've just heard. Biological energy largely comes from the sun, and it's produced there by nuclear fission of hydrogen to helium. But actually, there's another source of biological energy, and that's geothermal energy in the deep oceans coming from thermal vents. And that energy has been provided originally by radioactive decay and by residual heat that has come from planetary accretion when planetary bodies have crashed into each other. And as we now know, this source of energy supports abundant life in the floors of the oceans around these vents. And we're very good at harvesting this energy increasingly via solar panels, via windmills, and also via hydroelectric dams, because this is also a way of harvesting solar energy. So what happens is that the sun evaporates water from the oceans, that goes up and forms clouds. The clouds come to mountains, rain forms, and then we collect the rain in hydroelectric dams. And that water has potential energy, which can then be used in turbines to generate electricity. So these are all ways in which mankind has learned to harvest and use solar energy. And I think it's of great interest that by far the greatest component of biomass on Earth is plants. If you look at this picture, you can see that the green area far dominates all the other components to biomass. And look where animals are down in this bottom right-hand corner. So really we're quite an insignificant part of biomass on Earth. And the power of sunlight on Earth is 124 petawatts. And that produces 89 terawatts. A very small part of it is used for human food intake. But man is also very good at consuming energy. And in 2017, mankind was consuming 19 terawatts. We harvest solar energy on an industrial scale by growing crops. We then either eat the crops or we feed them to animals, and then we eat the animals. And then, of course, we retail the energy in the form of foodstuffs, in the form of meats and vegetables and grains. Now, let me come to how the energy in foodstuffs is released in the human body. So here is a typical human cell, typical apart from red blood cells. It's bounded by a biological membrane here. And it's estimated that our bodies contain 37 times 10 to the 12th such cells. And inside the cell is the nucleus, where most of the genetic evidence is to be found. And close to the nucleus is the endoplasmic reticulum, where the proteins of the cell are made. And then they're sent to their destination by a kind of cellular post office, the Golgi apparatus, which directs proteins to appropriate parts of the cell. And then interlaced between these structures are the mitochondria, where the energy-converting events take place. And the mitochondria are not as often depicted in textbooks some bacteria-like structure. They're actually fused together in networks that extend throughout the cell. So this is a skin cell here. This is the boundary of the cell. The nucleus is up here. And you can see that the mitochondria are very dynamic, and they're joined together And they're constantly undergoing fusion and fission. So if you watch some of these small bodies, they will go and fuse with this reticulum. And in other, so this is the fused state of the mitochondria, but in other points during cellular life, this reticulum breaks up into small vesicles. So for example, during a cell death, apoptosis, the mitochondria break up into vesicles. And this is an area of very active study at the moment, not fully understood. So let's look at the mitochondrion itself. So it's bounded by an outer membrane, and it has a second membrane inside, which is highly folded. You can see it going back and forth here. And these are known as the cristae of the mitochondria, these convoluted structures. And the inside of the mitochondria, inside both membranes, is called the matrix. And it's here that ATP is made, so we need to get ATP into the cytosol to make our bodies work, but the ATP is actually made inside the mitochondrion. And so it has to be transported out of the mitochondrion and made available to the rest of the cell. And so ATP is the fuel of life, as we've heard already. When it interacts with water, it releases energy and produces these two products, phosphate and adenosine diphosphate, ADP. And what the ATP synthase is doing, as you've heard, is putting those products of hydrolysis of ATP back together again. And ATP is required in all kinds of bodily functions, for example, in the contraction of a muscle cell, in the synthesis of macromolecules like proteins and nucleic acids. It's required for electrical impulses of nerve cells and so on. And each day, each of us makes our own body weight in ATP. So an average human being is maybe 60 kilograms. So each of us is making about 60 kilograms of ATP every day. But at any instant in time, we only have 50 grams of ATP within our bodies. So what we're doing is we're taking these ATP molecules, we're breaking them down, releasing the energy to do the things that the energy is needed for, and the products, ADP and phosphate, are being recycled, as I've explained already. And each day, each ATP molecule in our bodies is recycled 1,300 times approximately. So as you've heard, the mitochondria are made by machines. So we have proton pumps that harvest energy from foodstuffs. And then that's stored as a form of potential energy, rather like water is stored in a hydroelectric dam. And then a molecular turbine, the ATP synthase, uses that potential energy to make ATP. And since the ATP is being made inside the mitochondrion, we have to have a transport system to take it out of the mitochondrion and distribute it around the cells to biological sites where it's required. And then, as we keep saying, the reaction of ATP with water releases energy to do work. And the way it works is complicated, but actually at the heart of it, there's a very simple piece of chemistry, because the chemical energy that's being used in the breakdown of foodstuffs that we're eating, we're actually burning them in a controlled way using the oxygen that we breathe in. This releases chemical energy. So this would be the inner mitochondrial membrane. And that chemical energy is then simply used to translocate hydrogen ions, protons, from the inside space to the outside space. And this builds up an excess on the outside. And this is a form of potential energy that can then be coupled to the synthesis of ATP. And this is the idea that Professor Yoshida explained a few moments ago of Mitchell. So this is the so-called chemiosmosis. And so the mitochondria oxidize sugars and fats in foods and then store the potential energy in the form of hydrogen ions on one side of a membrane. And the turbines of the ATP synthase, now in mitochondria, the ATP synthases are put two together. They make dimers. And the dimers, you recognize the shape of the ATPase now, the dimers are at an angle of about 90 degrees. And those dimers extend in long rows, effectively into infinity, along the edges of the cristae. And if you remove the capacity by genetic manipulation, if you remove the capacity of the ATP synthases to form these dimers, then the structure of the cristae in the mitochondria collapses. It's something that we're studying actively at the moment. So this is the product of about 25 years of my life. I started working on this problem in 1978, and by 1994, we had the first hint of the structure, which then led to the demonstration, as you've heard here in Tokyo, of the an experiment directly demonstrating rotation in the catalytic part of the enzyme. I've gone on to characterize the rest of the enzyme complex, and then with by X-ray crystallography, I've built up a kind of mosaic picture, which is shown here. It shows the rotor turning in the middle and driven by protons going back through the mitochondrial membrane. And you can see the stepping action of the rotor. So it's not, in this part, it's not turning continuously, but it's stepping. Now, I won't say any more about the ATPase complex. You've heard a lot about it already. And I wanted to change topics, really, and talk about more human aspects of science. And I particularly want to talk about infectious diseases and resistance to drugs, because this is becoming a huge problem for mankind, and particularly noticeable in the upsurge in tuberculosis worldwide. And I won't talk about these other topics. I'll save that for another day, where bioenergetics are definitely involved. So I want to take you back to 1928 to this famous experiment by Alexander Fleming, conducted in London, where he had an agar plate with the bacterium Staphylococcus aureus growing on it. That's these streaks here. And a penicillium mold came in through the window and made this colony, and it was clear that it was exuding a chemical that was preventing the growth of a Staphylococcus aureus. And this was the discovery, this chance discovery of penicillin. So that was in 1928. And what did Fleming do? Well, the answer is nothing. And so he described his observations in 1928, but he actually did no significant follow-up experiments. His description just lay in the literature for 11 years. When Howard Florey and Ernst Chain at the University of Oxford decided to start to investigate the chemical properties of penicillin, and particularly to see whether it could be used in clinical medicine for fighting against infectious diseases. And in 1940, Edward Abraham, who was my PhD supervisor at Oxford University, he noticed that if you treated Staphylococcus aureus with penicillin, you could select resistant organisms. So before penicillin had ever been used in clinical medicine, Abraham observed resistant Staphylococcus aureus. And this sounded an ominous note, which is still ringing today. So it was only in 1943 that penicillin was introduced into clinical practice by Howard Florey. He went to the battlefields in North Africa and treated Allied soldiers with penicillin. Now, what is going on in the resistance of, in the emergence of resistance, is that in any population of bacteria, there are organisms that are sensitive to the antibiotic, but there are ones that are resistant. And the sensitive ones will be killed off, leaving behind the resistant ones in orange, and then they will proliferate and take over. And so in 1945, as you know, Fleming, Chain, and Florey were awarded the Nobel Prize in Physiology or Medicine for the discovery of penicillin and its curative effects in infectious diseases. Now, as I've said already, no sooner had penicillin been deployed in 1943 than clinical resistance was observed in 1946, so very rapidly. Sulfonamides had been introduced a little bit earlier in the 1930s, but by the 1940s, there were resistant strains. And you can go down this list of clinically used antibiotics, and it's the same story. No sooner has vancomycin, for example, been introduced in 1952, than 1 year later, clinical resistance was observed. And there's growing alarm because antibiotic resistance in blood infections are increasing. So these show data collected in England up to 2017. You can see there's a steady rise in the number of antibiotic-resistant blood infections. And in 2015, actually, I'm one of these people in this column here. I was seriously ill from a very minor infection. And the World Health Organization have drawn attention to this problem in 2014. So in the bright red dots, you see the death toll from particular conditions, from tetanus, cholera, measles, antimicrobial resistance, cancers, diabetes, diarrhea, and car accidents in 2014. And then the prediction of what will be happening by 2050. So by 2050, antimicrobial resistance is estimated to be of the order of 100 million, and it will far exceed the number of deaths from antimicrobial resistance will far exceed deaths from cancer today. And the burden in 2050 will not be spread evenly worldwide. Western Europe will be, and North America and Australasia will be relatively exempt. And the burden will be felt particularly in Asia and sub-Saharan Africa. And we know the reasons for the spread of antibiotic resistance. One of them is overprescription of antibiotics. Another one is the overuse of antibiotics in the livestock and fish farming industry, and then obvious things like poor infection control in hospital. And another major issue is there's been a lack of new antibiotics being developed. I'll show you in a moment. There's also a difference in the way that antibiotics are used in clinical medicine in different countries. So I've depicted some Western European countries here, and it shows that in Greece and France, antibiotics are used much more for treating people than in the United Kingdom, France, and Germany, where they're used more sparingly. And often in France and Greece, antibiotics are given prophylactically to prevent a disease, or they're given inappropriately when people have viral infections instead of bacterial infections. And so there's concern about this and an encouragement to doctors to prescribe fewer and fewer antibiotics and only use them when they're really needed. And then we come to this problem of the widespread use of antibiotics in farming. And here they are being used as growth promoters. They're being used in a prophylactic sense. The animals are not ill, and yet 70% of the antibiotics in the United States, again, I think this is in 2015, was consumed by animals. And the animals then create a huge well of antibiotic resistance, which then spreads into the human population. Another problem that I mentioned already is this discovery gap. So in the 1940s up to the 1960s, there was this initial surge in the search for new antibiotics amongst natural products, leading to the discovery of tetracyclines, macrolides, aminoglycosides, chloramphenicol, and eventually to the cephalosporins that were discovered by my PhD supervisor, Edward Abraham, and have been used in clinical medicine ever since. And after about 1965, nothing much happened. There were no significant discoveries from about 1965 to about almost 2000, when people began to wake up again and said, look, we've got to start looking for antibiotics again. And this led then to the introduction of these compounds into clinical medicine. Now, tuberculosis, as you will know, is a disease of the respiratory system. It can spread to the brain, kidneys, and bones. It's highly contagious via coughing. Symptoms are chest pain, fatigue, weight loss, and fever. And worldwide, today, 2 billion people are infected. And it's estimated that in 2017, 1.6 million people died from tuberculosis. And 60% of those who are untreated will die. And often there's a coincidence of tuberculosis with HIV/AIDS because, of course, those people are often immune compromised. So why is there a resurgence in tuberculosis? Well, first of all, or until recently, the regime for treating tuberculosis was very long. It would take 6 months. And many of the people with tuberculosis are living in rural areas of the world. It's very difficult for them to sustain a 6-month treatment. And then came this problem, not just of drug resistance, but multiple drug resistance, and then extremely drug resistance, and then totally drug resistant. So in India now, there are isolates of totally drug-resistant strains of Mycobacterium tuberculosis. And as I've said, there's a widespread synergy with AIDS. Diagnosis is not quick and it's difficult. And the drugs and vaccines available are outdated. And an additional problem is that while some of the drugs will kill off actively dividing Mycobacterium tuberculosis in the human body, the Mycobacterium goes into a cryptic state where it's surrounded by T cells and B cells. So these are the green rods of the Mycobacterium. They're surrounded by T cells and B cells and encapsulated in collagen in these granulomas. And in this state, the Mycobacterium tuberculosis is sheltered from the effects of any antibiotics, which will kill off actively dividing organisms elsewhere in the body. But these things will escape. And then there's a mechanism whereby the granulomas will leak due to the effect of a proteolytic enzyme. This leads to the release of these cryptic Mycobacterium. They'll start dividing, and then the disease will re-emerge in a patient that had been thought to be cured. So any effective drug needs to kill both actively dividing and Mycobacterium hidden in granulomas. And so we need new drugs against novel targets. The only vaccine to be used still in clinical medicine is BCG, Bacillus calmette-guérin, which was introduced in 1921 by the Pasteur Institute in Paris. And I mentioned already problems of diagnosis. Now, about 10 years ago, this paper appeared in Science, a diarylquinoline drug active on the ATP synthase of Mycobacterium tuberculosis. This drug is called bedaquiline. It was discovered by Johnson & Johnson in a random screen. They had no idea what the drug target was, but it turned out to inhibit the ATP synthase. And it shortens the treatment for tuberculosis down from 6 months or more down to a few weeks. And it's effective against both the dividing state in the bloodstream and elsewhere, as well as the cryptic state in granulomas. So it's the first anti-TB drug with novel action to be introduced into clinical medicine in 40 years. It inhibits the ATP synthase by binding to the rotor. So it stops rotation of the rotor here and stops the production of ATP by the Mycobacteria. And as I've said, it's active against the dormant and replicating states. Most importantly, it's active against multiple and extremely and totally resistant Mycobacteria. So this sounds wonderful. The drug is called bedaquiline, and its chemical structure is shown at the bottom. However, unfortunately, bedaquiline is not a wonder drug. FDA approval was based only on phase 2 trials. Now, anybody who's been involved in drug trials will know that that's unique, and that normally you'd need to go into phase 3 trials, and you'd also need to have treated many, many more people than 440 people before a drug would be released on the human population. It's recommended only for multiple extremely and totally resistant cases. There's a very significant cardiovascular risk associated with taking it. It's very lipophilic, and it persists in the human body for 5 to 6 months. Now that again, pharmacologically would be a totally unacceptable property, but such is the dire need of the people with tuberculosis that it's been allowed to be introduced into clinical medicine. And unsurprisingly, the patients are susceptible to this condition called phospholipidosis. And it can lead to death in the people that are being treated, and 10% of those deaths are unexplained. So although it's helpful in many cases, there are these very significant complications. And so the message is we need more inhibitors of mycobacterial ATP synthase. And so what my colleagues and I have done is to solve the structure of the catalytic part of the mycobacterial enzyme earlier this year. And this now serves as a target against which anybody can try and design new inhibitors that will inhibit the catalytic part of the enzyme. We're working on the structure of the entire enzyme. You can see we're making very good progress, and I expect this to be completed in the next 6 weeks. And this will contain the membrane domain where rotation is generated, and it will have bedaquiline bound to it. So we'll know exactly where bedaquiline bounds, and this will be useful to medicinal chemists in making variants of bedaquiline, which of course is what happens in the pharmaceutical industry. Now, we're also working on not the human ATP synthase, but the bovine ATP synthase. We've now solved the entire structure of the dimer that's depicted here. So each of these elements contains 29 different proteins. It's a very complicated structure. And in this dimeric state here, because of the flexibility of each of these elements, we've resolved 40 different conformational states, and we're just analyzing them at the moment. I don't think they have any importance for the catalytic activity, but actually what they're important for is the formation of the cristae in the mitochondria. So we're beginning to study how mitochondria are put together. So as I said earlier, I started this journey in 1978, and finally, 40 years later, I've managed to get to this point. We characterized all of the proteins in this complex. We started out determining their amino acid sequences. We did the same for bacterial systems. We crystallized the catalytic part. That took me 10 years to achieve that, which led to this structure you've heard about in 1994. And then we built it up, as I explained, in a mosaic, and finally we've got the whole thing. Now, I'm going to end there, but when I went to Cambridge in 1974, I went to work with Frederick Sanger, who already was a Nobel laureate, and shortly after I'd arrived, he won a second Nobel Prize. And he was interviewed by the British press, and it was quoted in the newspapers that Frederick Sanger said, "Most experiments fail." And this was really, it had two elements. One was that he, in his own experimental work, in working out methods for sequencing DNA, he'd encountered failure repeatedly. But what he didn't say is that when you encounter failure, you have to learn from failure, change your experimental conditions until the experiment works. So in other words, difficult experimental science requires persistence. And this reminded me of another quotation from Winston Churchill, and he said, "Success consists of going from failure to failure without loss of enthusiasm." I think that encapsulates some of Churchill's life, and it's something that I've learnt as well, is that if you can't cope with failure, then really you shouldn't be doing this kind of difficult experimental science. And the other aspect, which is strange from a personal point of view, is that when I was an undergraduate studying chemistry at Oxford University, the first research project I ever did was on Mycobacterium tuberculosis, where I extracted cell walls and synthesized branched-chain fatty acids. So there's a kind of circularity in all of this. I started out, and then I went to work on antibiotics with Edward Abraham. And now at the end, towards the end of my career, undoubtedly approaching, I'm now back working on antibiotics. So from a personal point of view, I found this quite interesting and very satisfying. So that's my story, and I hope that you've enjoyed it. So thank you very much for coming and listening to me this evening.